This video is going to look at carbohydrates, which is a biomolecule. They serve as energy stores and fuels in plants and animals, such as glucose, sugar, and starch. They're also part of RNA and DNA, as well as structural elements in plant cells, such as cellulose. Carbohydrates can be broken down into different categories. Monosaccharides are the basic units of carbohydrates, and they have between 3 and 8 carbons, multiple hydroxyl groups, and either an aldehyde or a ketone functional group. Disaccharides are two monosaccharides that are bonded together through a glycosidic bond, and polysaccharides are many monosaccharides bonded together through glycosidic bonds. Let's start by looking at monosaccharides, their structure, and talking a little bit more about how we can classify them. So as I mentioned before, they have between 3 and 8 carbons. They have multiple hydroxyl groups and either an aldehyde or a ketone functional group. So let's start by looking for those aldehyde and ketone functional groups. So if you'll remember that an aldehyde functional group is a carbonyl bonded to at least a hydrogen on one side. So here we have, I've circled the aldehyde functional group in this monosaccharide. You have the carbonyl group bonding to a hydrogen. So that is the aldehyde functional group. We can classify this monosaccharide as an aldose. When you see that OSE ending, that's indicating it is a carbohydrate. You'll notice that when you look at the names of a lot of carbohydrates, they will end with an OSE, like glucose or sucrose or lactose. Now let's look at the other one. Let's find our carbonyl group, and I'm going to circle the carbons that are bonded to it. So here's our carbonyl group right there, and it is bonding to carbons on both sides. And so because your carbonyl is bonding to carbons on both sides, it is a ketone functional group. And so we would classify this as a ketose. Okay, so you can classify your monosaccharides by functional group. You can also classify them by how many carbons they have. Both of these examples have 1, 2, 3, 4, 5 carbons. And so we would classify that as a pentose. Pent meaning 5. And so if you had 3 carbons in your monosaccharide, that would be a triose. You have that OSE ending. 4 carbons would make it a tetrose. 5 carbons is a pentose. 6 carbons is a hexose. Those are the most common there. Okay, and both of these are, if you count this one up, 1, 2, 3, 4, 5 carbons. So this is also a pentose. This one we had said was a ketose. And so we could say that putting it all together is a ketopentose. It's not the name of that particular monosaccharide, but it is how we would classify it. This is an aldose, and so this would be an aldopentose. Okay, another important thing about monosaccharides is that they are chiral objects. They are chiral molecules, which means that they have a non-superimposable mirror image. It's kind of like your hands. You have a right hand and a left hand. They are mirror images of one another, but they are not identical. You definitely have a right hand and a left hand. And so you have these right hand molecules and left hand molecules. And we're looking at right now what's called the Fisher projection for these monosaccharides. They're these straight lines. They are either vertical or horizontal, showing the bonds. But a couple of things about that. One thing is that you have to remember for carbon, those single bonds around carbon are going to take on a tetrahedral arrangement. You have multiple hydroxyl groups, and remember those the ones where the oxygen is bonding to like a hydrogen on one side and then the carbon on the other side, those two bonds have a bent structure. And so while the Fisher projections make it neat and easy to look at on a piece of paper, this isn't the reality of the 3D shape that these molecules have. That's the first thing to keep in mind. The other thing to keep in mind is that under physiological conditions, so the conditions that would happen inside your body or inside a cell, in a plant cell, these monosaccharides are going to be, for the most part, in ring structures. So they are not these straight chain molecules anyway. But we're going to look at these Fisher projections because they make it easy to look at. And I'm going to go through the process of figuring out how you would classify it as the right-handed molecule or the left-handed molecule. And so one thing you need to do is find the carbonyl. And so on these, the carbonyl, usually in Fisher projections, they're written towards the top. So there's my carbonyl. Now I want to find the chiral carbon that is farthest from that carbonyl. So how do you know which carbons are chiral? Well, a carbon is chiral if it is bonded to 4 different groups. And so usually your middle carbons in your that are not carbonyl groups are going to be chiral. The very bottom carbon in a Fisher projection, this one down here, is not chiral because you have 2 hydrogens right there. So it is bonding to 2 identical groups, 2 hydrogens. But we are looking for the carbon, the chiral carbon that is furthest from the carbonyl. In this one, on these, it's those carbons right there. And then you look to see which side is that hydroxyl group on. When you are looking at the molecule, is that hydroxyl group on the right or the left? And so for this one right here, that is on the right side. And so we would say that this one is the D isomer. D being for right, dextro in Latin. And on the other one, this hydroxyl group over here is on the left side. So this would be our L isomer. So that's easy enough to remember. L stands for left, and so then the other one, D, is going to stand for right. Now, why does this matter whether you have a D or an L isomer? Well, D isomers are the ones that are most commonly found in nature, and so your body for sure can tell the difference between the D isomer of a monosaccharide and the L isomer, and it's going to use up and produce these D isomers. Okay, I want to get out of PowerPoint and real quick and show you in another program the 3D view of those 2 that we were just talking about, that we looked at the D and L isomers of. And so I can spin these molecules around, and I want you to see that these that those bonds are not in a straight line, that they are at angles because they have that tetrahedral arrangement around it. And so you can see that around this, the second blue molecule from the bottom, or the second blue atom from the bottom, is our chiral carbon. And so you can see that you have a hydroxyl group, which is the oxygen is red, coming out towards you on one side, and it's on the other side coming out towards you from the other molecule. And so they are mirror images of one another. They are not superimposable. So this gives you a little bit of an overview of the structure and how to recognize different parts of monosaccharides.